Now we look at the class Epsilon-Proteobacteria, the order Campylobacterialis, and under this order there is two families of interest, Campylobacteriaceae and Helicobacteriaceae. So under Campylobacteriaceae, Campylobacter jejuni that causes uh, enteritis, and Helicobacter pylori that causes peptic ulcer are important. So Campylobacter is a gram-negative, microaerophilic, motile with single polar flagellum, oxidase positive, and spirally curved bacteria. So an important point here is it adapts well to intestinal environment of poultry, where domestic and wild animals are reservoirs. Poultry is an important source of infection for Campylobacter gastroenteritis. So Campylobacter gastroenteritis is usually caused by Campylobacter jejuni. Although there are also Campylobacter fetus that causes bacteremia, but only in mostly compromised hosts. So bacteremia is also known as blood infection. Healthy individuals, Campylobacter fetus only causes self-limiting diarrhea. Campylobacter jejuni incubation is about 2 to 5 days. Illness lasts less than 1 week. It causes and then produces an enterotoxin that's similar to the one from E. coli. It is an acute gastroenteritis with fever, cramping, abdominal pain, diarrhea, dysentery, and vomiting. Diagnosis: it requires microaerophilic and capnic conditions, an increase in CO2 concentration, and a reduction in oxygen concentration. So culture on blood agar plates is adequate, and treatment is available with macrolides or fluoroquinolones. But about 0.1% of the case will develop a autoimmune disease, like the Guillain-Barré syndrome, where there is temporary paralysis because there's an autoimmune attack on the lipids of nervous tissue. The other important family in the order Campylobacterialis is Helicobacteriaceae, with an important genera Helicobacter and the type species Helicobacter pylori. Helicobacter pylori is spiral-shaped, multiple flagella, and it's a microaerophilic bacterium that is responsible for most cases of peptic ulcer disease, including gastric and duodenal ulcers. So it is first cultured by Robin Warren and Barry Marshall in 1982, and this discovery won them both the Nobel Prize in Medicine in 2005. So both of them are from Australia. Barry Marshall was the student and Robin Warren was the supervisor. So they saw bacteria in the stomach lining of endoscopic samples taken from patients with ulcers. Because stomach has acidic pH, not many thought that bacteria is able to survive in the stomach. Therefore, not many researchers took interest in these observations. However, Barry Marshall and Robin Warren continued pursuing this, and their first major breakthrough was when they were able to culture the bacteria from the samples. So once they were able to culture it, consistently from ulcers. The next stage in to fulfill the Koch postulate was to inoculate a healthy host to see if the same symptoms arise. Unfortunately, at that time, there was no animal model suitable for Helicobacter pylori. So Barry Marshall drank a flask of Helicobacter pylori, and sure enough, he developed symptoms, and samples were taken that shows the same bacteria in the stomach lining. Therefore, from this observation, from this experiment, they were able to prove Koch postulate that it is Helicobacter pylori that causes peptic ulcer disease. Although this discovery is very important, there were a lot of resistance from pharmaceutical companies because the discovery that ulcers can be caused by bacteria and therefore cured with antibiotics is against the detrimental to the profit margin of these pharmaceutical companies. However, now we know that other host factors are involved because only 15% of those infected with Helicobacter pylori actually develops ulcers. Many infected show no symptoms. So Helicobacter pylori actually can penetrate the mucus layer lining of the stomach and can grow in the highly acidic stomach. It is able to do this because it produces a large amount of urease. This urease will convert urea to ammonia and thereby increasing the pH around the bacteria. As it neutralizes the pH around its environment, the ammonia that is produced also destroys the stomach lining, facilitating the development of ulcers. The ammonia produced is toxic to the stomach lumen epithelial cells. Other enzymes, for example protease, catalase, and phospholipase, also cause cellular damage. So together with the CAC-A encoded protein, the gastric epithelium will be injured. At present, Helicobacter pylori is also designated as a carcinogenic bacterium, as 3% of those infected with Helicobacter pylori actually will further develop into gastric cancer. So for diagnosis, tissue biopsy and culture of the microorganism is recommended. Breath tests for 13 carbon labeled urea and 13 carbon labeled CO2. The expelled air is also available. And for children, the tests for Helicobacter pylori are the antigens in the stool. And for treatment, you have the Pepto-Bismol and metronidazole that can remove the bacteria. However, at present, now metronidazole resistant Helicobacter pylori strains have also emerged.